Afternoon, everyone, and welcome to assignment 4. This is an video recording the summation of assignment 4 for the class CS800 Research Method, Spring 2026. In this assignment, what I have done is I have read 5 papers related to my research and produced a literature review, showcasing my understanding of this current research in the field that I am currently working in. Basically, in this literature review, I have include contains these 4 sections: introduction, summaries, synthesis, and references. And the 5 papers I have used are paper 1, SCBOT, a large-scale pretend deep learning model for cell type annotation of single-cell RNA sequence data. Basically, this paper introduced the concept of building a foundational model that can be trained on a large amount of on-label dataset in a safe, supervised manner so that it can later be fine-tuned on a specific task such as cell type annotation. It uses bot transformer model for the single cell genomics. On this, another paper supplements this. The paper name is Transfer Learning Enables Prediction in Network Biology. What it basically does is building another model called GenFormer, which is also a bot transformer model. What it does is it scales the data, the previous training dataset, and also scope of the downstream tracks, such as not only cell type annotation, but perturbation predictions, batch integrations, and so on. And the next paper that also supplements the previous one, the GenFormer model, is SCGPT, towards building a foundational model for single-cell multi-omics using generative AI. This paper basically does is allows multi-omic dataset of single-cell multi-omic dataset, not only single-cell sequencing data but also protein data. So to train the foundational model so that it can be fine-tuned on uh, downstream tracks such as batch integration, perturbation predictions, cell type annotation, and so on. Uh, and Another paper I have used is deciphering driver regulators of cell-fate decision from single-cell transcriptomic, transcriptomic data with Silicon. What it basically is giving the idea that the transformer model or other model uh, are not sufficient for certain tasks that the single cell genomics or how the biology works required. For example, creating a gene regulatory network to find the driver regulators of the cell fate decision. So it basically uses a graphical neural network with attention mechanism to overcome these issues that we cannot solve or basically is not uh, structurally in applicable with the transformer. And another paper I have read is I have included SC Mamba, a pretend model for single cell nucleus RNA sequencing analysis in neurodegenerative disorder. This paper basically supplements the previous paper 1, 2, and 3. These 3 papers basically suffer from the transformer bottleneck problem, of which has which in which as the sequence length increases, the computational or memory requires increases exponentially with the power of n square. So this basically, SMMA basically uses state space model, such as a Mamba model to be particular, for the uh, as a foundational model for training single cell sequencing data so that to overcome the bottleneck problem that the transformer has. And these are the directive for the assignment. And I have used the LaTeX for creating the uh, literature review. And this template is being provided by the uh, professor of our CS800 class. And in this literature review, you can see we have introduction, summaries, synthesis, and references. Introduction is a basic background of how these papers solves what the problems are and how these papers or research papers solves those problems. Summaries provide what are the contributions of each paper, and synthesis is basically how this whole paper connect together and is relevant to my research area. So you can go through all of this, and these are the references paper are basically the papers for the These 5 papers I have read, and this is the give tech for all of the reference papers. These files are also included in the assignment submission. Thank you.